But thank you so much for joining us for Bugs Brews tonight. I'm Glenn Morangelo, and I'm one of the founders and the development director for the Missoula Butterfly House and Insectarium. And tonight's the second Bugs Brews lecture of 2025. And it's the second lecture that our friends at Missoula Community Access Television, better known as MCAT, are recording as part of a media assistance grant donated to the Missoula Butterfly House and Insectarium. And we're thrilled to be working with MCAT to begin documenting some of the fantastic speakers we're fortunate to have as part of this lecture series. This is only the second one. We've got years of other folks who it just has dissipated into the ether, but we'll start getting these up online. I don't believe we do have a link on our website yet to be able to watch it, but I checked the MCAT website today, and if you just go on there and hit their little search icon and type in Bugs, it'll come up. So you can watch the first one that was recorded last month with Dr. Eric Green. So thanks again, MCAT. Speaking of saying thank you, I saw a couple of our draft work friends are here this evening. As always, a huge thank you to our friends at Draftwork for generously donating all of the beer and the hard seltzer for this evening. And not only that, and not just this evening's lecture, every lecture that we've had since we started this lecture series about 9 years ago, I believe it was. So Draftworks has been incredible to us over the years. For those of you that have been involved in nonprofit organizations and doing events like something like Bugs Brews, you already know how important these kind of programs are as far as education, but also for community engagement. And I kind of like to refer to these events as friend-raisers, because yes, you make some money and you more than cover your costs, but the primary focus is on education and connection. But thanks to our friends at Draftworks, since they donate all the beverages, we don't have to buy the beverages for this. It actually is a little bit better fundraiser than it normally would be for us, so we thank them tremendously. Speaking of beer in cans, just a little housekeeping. Yeah, cheers to everyone, thank you very much. But speaking of cans, we do have 2 recycling bins on either side of the beer window. And I just ask that if you have not finished a beverage, please do not throw that into the recycling bin and just put it on the counter and we will deal with it. Because last time there was a pretty good swamp that we had to go through at the bottom of the bucket. Okay. Wow, I'm already at the highlight. No offense. The highlight of the evening, the joke section. As everyone knows, I always tell a couple jokes. And I've been telling jokes at events for a really long time, and I'm definitely starting to recycle some of them, and I'm really starting to dig down into my B material. So, and BEE material. I said that joke a while ago. But here goes nothing. What is on the ground and also 100 feet in the air? A centipede on its back. All right. And I figured this was one that I found a while ago that I just thought I'd try. What is green in color, has 6 legs, and can kill you if it falls out of a tree and lands on you? A pool table. So not you thought I was going to talk about insects, but no. All right, just really quickly, some plugs for upcoming events. This Saturday we've got Curious Creatures, and this is kind of a family group education program, and this month is going to be Beetlemania. It'll be focusing on what all the 400,000 plus beetle species have in common and how they adapt to survive in various habitats. So if you've got a family group or just a group of friends that want to learn about bugs, it's this Saturday from 10:30 to 12, and you've got admission to the Butterfly House and Insectarium before and or after. On Sunday, we certainly are filling in the programs here. I think this is the 3rd time that we're having this, but we're holding a relatively new event, which is a guided meditation in the Butterfly House from 9 a.m. to 10 a.m. It won't be a 1-hour meditation, but I believe it's a 10 or 15-minute meditation with time to enjoy the butterfly house. There's still a few spots available. March 18th will be the next Bugs Brews, and we will be hearing from our very own Rob Taylor. And he will be talking about the co-evolution of flowering plants and butterflies. If we get all of our stuff together, tickets should be going on sale this Saturday at 9 a.m. So what we're trying to do is get in the habit of when we have Bugs and Brews, the tickets for the next one will go on sale that next Saturday. Monday, March 24th, we'll be holding our adult quiet hours from 9 a.m. to 10 a.m. No kiddos, just adults for this kind of library quiet time in the Butterfly House. And then roosting hours on March 27th. It's basically like Bugs and Brews, but without the lecture. There's some beverages thanks to DraftWorks donating to that as well. And it's just adult time in the Butterfly House. We also offer crafts. So it's always fun to see adults get into crafts that they haven't done probably since they were in elementary school. And some trivia and other things like that. And I think that's all I wanted to talk about, aside from introducing our speaker this evening, Mat Seidensticker. Tonight, we're really excited to welcome Matt. He's with the Northern Rockies Research Educational Services. Matt will be talking about the Montana Moth Project, introducing us to the dark side, the amazing world of moths. Matt developed broad interest in wildlife research and natural history while earning a wildlife biology bachelor's in 2000 and an environmental studies master's in 2011 at the University of Montana. After his undergraduate studies, he worked with the Owl Research Institute for over a decade, conducting field studies on snowy, long-eared, saw-whet, flammulated, pygmy, and northern hawk owls in Alaska and Montana. His graduate research on flammulated owls and subsequent work with the avian science team at MPG Ranch ignited a passion for moths and insects, leading him to establish the nonprofit organization Northern Rockies Research and Educational Services in 2019 to advance scientific research, monitoring, and conservation of moths and other insects in Montana and beyond. Moths are arguably one of the most ecologically important insect groups in terrestrial ecosystems. However, they tend to really fly under the radar in terms of understanding about them, their diversity, distribution, and habitats in Montana. So tonight, Matt will discuss how the Montana Moth Project started. He will give us an update on its current progress, and we'll discuss its future direction. So with that, Matt.
Thank you. Can you guys hear me? Is that good? Yeah? All right. Well, I'd like to start just by extending my gratitude to Glenn and Jen and the Insectarium for inviting me to speak tonight. It's just a real privilege. I'm really excited to be here, talk to you about moths. And there's my wife. I'm going to find my wife here. She's going to be my moderator tonight a little bit because I can really get going. So I'm going to try to keep this to 45 minutes. Partly because I have a lot to talk about and there's a lot of exciting stuff to talk about, but probably mostly it's what my mother used to endearingly tell me, and I'm sure Peggy would agree that there's a reason my eyes are brown. So I'll try to keep this, to the point and not get too off track. And so, like Glenn mentioned, I'd like to talk to you about the Montana Moth Project tonight. And the Montana Moth Project is our flagship program for our little nonprofit. And it's essentially an effort to survey diversity and distribution of moths statewide. And doing that by surveying moths in each of the 56 counties in Montana. And so I wanted to talk to you about how it started, where we're at, and where we hope to keep it going for. And so the origins of it really go back to barcoding and bird poop is what it goes. And some of you might know the Insectarium has done a little DNA barcoding. DNA barcoding is basically using short gene fragments in the mitochondria generally for species identification and things. And it's the cytochrome oxidase 1 gene. And usually they're so conserved within a species but variable among species that you can literally take a sample from like a tissue sample, have the DNA sequence, and be able to tell what species it is. So that's just DNA barcoding. So basically, you're taking a sample. This is a sample plate with like a leg of a moth in there. And then you're amplifying the DNA, and then you get a species identification. DNA metabarcoding, it deals with where you have like a mixed sample. So let's say you have a malaise trap, for example, you have a bunch of insects in it, or you have an environmental sample, whether it be like water or soil. And in this case, bird poop. We can literally take those samples, amplify the DNA in those, and then determine what those sequences are. A lot of times to species level. And so as Glenn mentioned, I was working with the MPG Ranch, Kate Stone et al. In 2015, and they had started a project on poor wills and nighthawks and bats and things. And initially we were just kind of tracking them around, trying to find their nests, their activity patterns. But in 2017, the light bulb kind of went off, and we were like, well, we really want to know, because we were finding these poor wills, these little guys here, are, we call them pellets. So these birds defecate these really, they're about the size of a dime, let's say. And they really hold up well in the environment, and they don't break down. And so we thought, well, geez, they're nocturnal, right? How are we going to figure out how these things, what are they eating? And so we're like, well, why don't we try this barcoding thing? And so we started collecting the bird poop. And one of the things we really learned early on is that these sequences that are generated, you need a reference sequence to compare it to get these identifications. And a lot of the reference databases that are out there, even though like the Barcode of Life database and things is really large and has millions of sequences, they really lack the taxonomic coverage in a lot of reference sequences, especially for insects. So we were like, well, why don't we build our own reference library to compare that to? And so I went out in earnest in 2017 and 18, started setting up light sheets, collecting, anything that came into the sheet, whether it was a moth or a beetle or wasp, whatever it might be, and we started doing this barcoding to build a library that we could use as a reference. And then in 2019, we expanded that to more of a ranch-wide effort, and we ended up with this barcode library. And you can see here a lot of those flies, but there's wasps and things. But I only show you this because this lepidoptera here, the number to look here is bins. And so the bins are they're kind of proxies for species. And so they're called barcode index numbers. And so they're closed groupings of really similar sequences that can be used as proxies to species. And so the first 2 years were pretty minimal effort. And that's Lepidoptera, and most of those are moths. And so we discovered, we barcoded like over 800 species of moths on MPG Ranch. And it was just kind of like, wow, I had this casual, interest in moths with my grad research, because flammulated owls eat moths and things. But I really had no idea that it was such a grand diversity like that. And so this all culminated in a study we did. But it was during that time that as I was like kind of validating these identifications and things and looking online at these resources like the Moth Photographers Group and things, I really began to notice that a lot of times there would be either no dots on the map, or there'd be dots, in all these other states around us, or there'd just be one single dot in Montana. And so I was like, geez, this is crazy. Like, how can this be? And it didn't take me long, and I started investigating, well, what's going on with moth research in Montana? And it didn't take me long to discover that single dot on these Moth Photographer Group's maps were from Bob Martin, who I call the godfather of Montana mothing. And he, for the last 20 years has contributed tons of photographic records of moths outside of Rimini, near Helena. But there really wasn't anything else like going on in terms of like structured research, or we didn't know about the distributions of like some of these moths Bob was finding, like where do they occur all over the state or just right there, things like that. And so in 2019, late 2019, early 2020, I started uploading a lot of our records to websites like iNaturalist. In this case, it was LEPS by Field Guide. And I got an email from a fella, and he's like, hey, I noticed this moth, this Sympestus socar. He's like, do you mind if I use that record in my season summary? I was like, oh yeah, for sure, yeah, feel free to do anything like that. Well, that fella turned out to be Chuck Harp. Chuck's a lifelong lepidopterist. He was, he spent his 40 years in retail, actually. He had a family to support, and like any wise man, he knew that he wasn't going to be able to do that being a biologist or an entomologist, and so he spent his whole career literally in retail, but at the same time, he's the Rocky Mountain Zone coordinator of the Lepidopterist Society. He's on the executive council, and he's a Shinnia moth expert. So he's worked out the food plant associations, host plant associations with Shinnia flower moths, and he basically was the guy that the Smithsonian went to learn all this information, and he was able to, like, you know, discover new species. And then when I received that phone call, he was working for the C.P. Gillette Museum of Arthropod Diversity in Fort Collins, in Colorado State. And so he said, well, by the way, we don't know hardly anything about moths in Montana. And I was just like, at that point, I was like, it kind of validated what I was thinking, right? And so I'm like, oh, okay, this is great. I said, well, by the way, I'm going to be doing a little study on MPG Ranch. And so these yellow icons are MPG Ranch, so I had some monitoring things set up. And so we started talking, one thing led to another, and I was like, okay, great, why don't we just start then with these 4 traps, and then these little icons here, we'll add those in. We'll set traps once a month. I'll collect the moths, I'll send them to the museum, and we'll just start there. Maybe 3,000 or 4,000 moths, right, in the summer. Well, myself and my tech at the time, we like to travel around and stuff, and so Carrie Boss was her name, she's like, well, do you mind if I take a moth trap out? And so she was camping in various counties, and pretty soon we were turning up like first state records and things like that. And so the 3,000 or 4,000 moths turned into about 10,000 by the end of that first summer, and I'm not sure if that's a look of excitement or exasperation or... And you have to remember, when we started this project, it was during the height, 2020, 21 was the height of COVID, so Chuck was basically the only one in the museum doing all this work. And so, we were able to send him a lot of moths that first year. And it was, towards the end of that summer, we got on the phone again, and we were like, you know what? We're like, if not now, when, right? We're gonna have to, why don't we just do this in every county in Montana? And so, that's it. That's how it all started. And so we said, okay, the first 5 years, we're going to try to do at least one sampling in each county. And then, years 6 or 10, we're going to go back and try to get a different habitat and things. And so now we're in our 5th year. Just completed our 5th year. We don't do it alone, though. In late 2020, Chris Grinter, who's the collections manager at Cal Academy, they were looking to expand their micro-lep collection, and so we started sending him a lot of the small moths, and then Marion Kirst reached out. She's an entomologist based in Billings. How can I get involved in this? And we'll talk more about her and the fantastic job she's done in growing the project over in the eastern part of the state. And so before we go too much further, I just want to take a quick minute to just, maybe not so much justify, but just be transparent and talk about why collecting, right? Especially in this day and age with loss of biodiversity and all these platforms of iNaturalists and taking photographs and things. I just find it really helpful and useful to just be upfront and transparent about that. And so the biggest thing is verifiability, right? So like photographs, it's true that we can identify a lot of moths based on a photograph. And it's true that if you take a photograph of a moth and Chris Schmidt or Chuck Harp or some of these crowdsourcing, if 10 people say that's what it is, most likely that's probably what it is. But in the strictest sense of the word, photographs can't be ever truly verified, right? You need an actual voucher specimen to do that. And I just want to show you a couple examples here. So we've got these 2 moths here, right? If I were to show these pictures to an expert or even any of you, and you went and looked it up, we'd all agree that it's an Admetavis species, right? But is it a species singular or a species plural, right? And so based on this, you might say, okay, and the lighting's not great. Maybe this one's a little bit lighter, and maybe the orbicular spots are a little different. And there's some things here that you may be able to say, oh, yeah, I can't understand. But that could be attributed to sexual variation or just regular old phenotypic variation, kind of things like that. And so if we look at it spread, right, here's where you can see maybe this one's a little darker, and you look at these spots, they're a little bit different, right? And maybe these terminal lines are a little different. But those really aren't stable characters, right? Because they kind of they vary. But if we look here on the highway, right here, and you can probably see that from where you're how it's concave. This is more or less straight, right? Right, so this is a stable character that we can use. And so this one here is a Metavus oxymoris. This one here is a Metavus icarus. And that's a newly described species. This one was just described in 2008. And they both fly together in similar habitats, right? So off of these pictures alone, you may not be able to, you probably just say, well, maybe we're in this place and there's records for oxymoris, but no. So that's the kind of the challenge. And here's another example, and this took me like 4 years to figure this out. So these are spilotus, which are really, really common in rangeland habitats, grasslands and things, even around here. One species or two species, right? And I struggled with this for years. Like, this one's supposed to be like a little smoother, the other one's a little more patterned, right? But they vary and stuff. Well, it took me 4 years to figure this out. If you flip them upside down, this has a really distinct dark spot and a nice clean hind wing. This is sort of dirty, right? So that's Spilotus clandestina. That's Spilotus bicava, right? And so that's why we collect, is to collect this material so that it's truly verifiable and so that it can truly be repeated. And the physical specimens, they provide DNA, pollen, things that photographs can't provide, right? And so we abide by the collecting statement of the Lepidoptera Society. And we really try to we're just doing samplings, we're not repeating samplings, depleting populations. Interestingly enough, we find that most of the moths we catch are males. And I attribute that to just the fact that the females are the ones on the host plants, calling with the pheromones, and they're probably not moving around the landscape as much. So, and then the data sharing, we'll talk about that a little bit more. So we try to do it ethically, and this is just a little excerpt from a permit I had to write once. And so what we're really trying, it depends on your objectives, right? So what we're trying to do is we're trying to build a voucher collection that will not only provide this material for taxonomic revisions, and maybe we'll be able to go back and get some pollen off of it and looking like that, but that somebody can then, as a truly verifiable collection, that maybe 50 years from now, somebody could repeat what we've done and look at how things have changed with climate change or things like that, right? And it's truly verifiable. And so anyway, that's a long way of saying that's why we do it. Now this is we started in 2020, right? We added these counties, and in 21 we added a few more, and then 22, 23, we kind of progressed. And then I'm happy to say that as of last August, we were able to achieve our goal of one sampling in all 56 counties. And you'll notice that, there's clusters here where I live. Thank you. There's clusters here where Marion is based. And then there's this smattering of things. I did a little study on the Bison Range, and then we're doing some work in Glacier. But as you can see, Montana's a really big state. And so we've got a lot of work to do. But it's kind of nice, yeah. Glenn was asking me how many miles I travel, and I average maybe 5,000 or 6,000 miles a summer. Some of these trips, when I go back, the eastern Montana, it can be 1,500 miles by the time I get back home. But really cool. And so what we try to do is not only sample like the most common habitats, but we've also tried to target like some of Montana's more unique habitats. This is the Pryor Mountains foothills. So this is, and we'll talk a lot more about this later on, because this is a very special place. It's our only true cold desert habitat in Montana. It's unglaciated. It's kind of at the nexus of 3 ecoregions. It's just a really special place. And then this is our light sheet, and you can see the trap off there. And so Protogygia is a genus of moths that was really poorly known from Montana before we started sampling there. We found 3 or 4 species in the Pryors, and now we've found it across the state. The Centennial Sandhills, the Nature Conservancy property in the Centennial Valley, believe it or not, these are all sand dunes underneath here, stabilized by vegetation. And so we see a specialist species there. This is a dune obligate, Copolepharon. We've found those guys there. We've tried, we've looked at pine butte fen, Turned up a pretty cool moth there, Hippocyna vagus, which is sort of a wetlands kind of specialist moth. The Bitter Creek Badlands up north, actually it's northwest of Glasgow. Really beautiful area. We've turned up some, some of these badlandy moths there, this Rhizogratis stylata. The Ross Creek Cedars, which is kind of just up the road. We've done some work there. Symphistus youngi, which is kind of associated with these more music forests kind of thing. The acid shale forests in central Montana, really another unique area. I'd really like to get back to there. Uzoa oberfolii. And so we're trying to get, the predominant habitats, but also some of the specialized habitats. And so now when we return in the next round, we'd like to maybe repeat some sampling in some of these habitats, but in different times of year. But also like in our mountainous areas, we're trying to get like a high point and a low point, right? So like maybe somewhere at elevation and somewhere down in the valley kind of thing. And then back east, it's places like this, like grasslands, but then these little pockets of unique little habitats. And so how does this all work? You kind of saw the slide of, we set up the light sheet, which is a white sheet with mercury vapor light. And we usually use like 3 or 4 bucket traps that use UV black lights. And we set those at like varying distances from the light sheet. And then we collect the moths. So how do they get from our buckets to the museum, right? Well, this is a thing, we use these freezer containers that you put like lasagna in or whatever, they seal up really tight. And we make what we call moth lasagna, right? So this is a layer of like surgical cotton. Put a layer of moths, put a piece of tissue over that, another layer of cotton, another layer of moths, and we can get like 4 or 5 layers in a container, which, there's maybe 25 to 40, depending on the size of the moths. So let's say, 150, 200 moths per container. And then in the field, we put those in coolers with dry ice to preserve them, keep them nice and fresh. And then they get shipped down to the museum where they get prepared by spreading, right? And so they do the real work down there, the hard work. Is they do all this spreading, and these, this could be all from one site, but 10 to 12,000, yes. Oh, so we use, it's called ethyl acetate. And so we use the little stubby water bottles with a cotton wick. And think of ethyl acetate as like it smells like a high-powered like nail polish or something kind of thing. And we just suspend those inside the bucket. And then the fumes, when they come in, the fumes euthanize them. And it's amazing that I guess it's kind of it's almost like it suffocates them, because we really have to, like in the morning, plug those holes, because they can detox it really fast, and they'll wake up like an hour later if you like. So it's the most humane way. I mean, some people use like cyanide and things, but it's really the most humane way that we can really do it. They get spread, right? These 10,000 moths or whatever that we send down a year, they get spread, and then they get put labels on, right? And our labels are really different. Oh, nice, okay. Jeez, okay. So then these little labels that then go on each. So if this is all one site, and this is one site, these labels get cut, put underneath the pin. And then from there, they get sorted, they go into mixed drawers. And so each site kind of just gets thrown in these entomological drawers, kind of all mixed up. And then we go through those mixed drawers, and then we sort them into morphospecies and try to put names on them, what we think they might be. And then from there, they go to the database. And we database our stuff in the thing called ecdysis.org. It used to be scanned, but this is all publicly accessible. So if you went to ecdysis.org and you go to the search function and then pick CP Gillette Museum and type like Lepidoptera Montana, it would come up with all the records that we have, and we'll talk about how many we have in there at this point. And this is kind of like, we try to take like exemplar pictures of each taxa, especially if they're really good. And so not all the records have pictures, but it all has all this metadata here, where you can see where we collected it, who collected it, basic information about the habitat, things like that. And then from there, once they're databased, they get curated and organized. And so here's a, curated box of, I don't know what those are. There's like a Pammy or something. But then they get header labels that get put in them, and then those go into these cabinets. And so these are all period drawers, and these are arranged taxonomically, starting with micromoths and the Saturniids, sphinges, geometrids, and then all the noctuids. And so we have 7 more cabinets on the back side of it. And so I mentioned that, we get the fun part. We get to just drive around, set traps, collect moths, send them moths. The museum is where, the amount of hours and work that they do, it's just incredible. And we've been really fortunate that Chuck has built a very vibrant community, and we have such an awesome support from the administration down there. So many people have been excited about the project that we've been able to open up opportunities for students and things like that. And so this is just kind of a wall of all the volunteers, students, paid or unpaid, whatever, from the museum. There's about 40 of them. This is a shot of the most students working in the collection at one time. And then this has kind of been our core sort of group. There's a couple missing here, but these folks have been working. Jan is a volunteer that comes in databases. She's been working on the project since it started, database of Montana Moths. And then, some spreaders and things. And then one of the folks that it wasn't in that picture, and I'd be remiss if I didn't mention him, is Greg Pankowski. He's a retired attorney that drives 2 hours to the museum once a week, and he has databased thousands of Montana moths for us. And so I owe a lot to Greg. Here's just an action shot of Jolie, pinning, and this stack of pins is usually empty by the end of the summer, stocking the boards, and then Olivia doing some sorting and things. And so we're just super fortunate. None of this would happen if we didn't have that repository and a relationship with Colorado State to be willing to do this. And it's been really, like I said, it's been really great because a lot of students have had opportunities to learn how to spread and database and learn moth identification. And we do our best to try to help, raise money to help support that endeavor. And so at this point, it's a little bit like drinking out of the fire hose. Like Chuck likes to say, it's like trying to change the oil in a car while it's moving. It really doesn't work out too good. But currently, this is just some brief numbers I pulled off of the ecdysis. And this is just for the quote-unquote macro moths. And we'll talk about maybe the difference between micro and macro moths here in a minute. But as you can see, some noctuidae, these are families. So the noctuid moths, we've got about 800 species almost, and then the geometric moths are around 300, and then so forth. Like these are tiger moths, and these are like your hawk moths and things like that. And so this just kind of represents the number of genre or genuses that are so 780 species out of 188 genre. These numbers are fairly close to what we might expect. So we kind of came up with a number before we started the project by looking at these distribution maps of how many species we might expect. The one, we're a little short on, quite a bit short on the geometrids. We expect about 450, so we're about 150 short there. And the noctuids, we haven't done a great job of doing some of the day-flying moths, and so we've probably got 100 noctuids. But all in all, we're about 36,000 specimens that encompass all the 56 counties. We've currently got about 30,000 of those databased, and that amounts, and this is just these families, about 1,283 species. That doesn't take into account like some of the subspecies and things like that. So we expect that number, we'd expect about 1,450 or something, so we've still got a couple hundred more to go to really feel like, but also, you'll also see as I go through these next slides that our definition of what to expect has kind of been blown up, because we've turned up some moths that you probably wouldn't have expected based off of the current distribution maps. And so with that, here I'll just show you a few examples of that. So this is one we found in the priors early on, the Shinya snow eye. It's one of those flower moths I talked about that Chuck has worked on. These guys are really, really like, not just like a plant family or a plant genus, but like a plant species specific moths. And so this one in particular feeds on wild onion, allium. alley wild ending, yeah. And so we've turned that up now, and you can see a sort of a Midwest kind of moth, a southern, Southwest kind of thing, and we're finding it up here. I think the Priors is the only place we've found it actually so far. But this Simpistis levis, these are kind of like, they're tend to be associated with like drier habitats or badland habitats, but we see them in the fall. They love rabbit bush. And so again, maybe one of these southwestern moths, and these are all our dots that we found them in now. Another Simpistis, kind of the same thing, right? And we're starting to see them in Montana. And then here's a good one. So we're really turning up even the eastern things. So this is Basilophia anguina. You can see this, and just to take these maps with a grain of salt, right? Because these maps don't mean that like there hasn't been somebody on a naturalist that maybe has photographed this thing, or maybe it's in somebody's personal collection. So this is just, or maybe the moth photographers hasn't uploaded. And these come from Moth Photographers Group out of Mississippi State. They've kind of been considered the official clearinghouse of moth records. And so these maps, just because it's not over here doesn't mean it's not, because it's on the map. But we're now seeing them in 2 counties. We've had this, just 2 specimens, but in Blaine and Choteau counties, I believe. But kind of an eastern thing, right, that we're now seeing in Montana. Another one of those kind of eastern things is Epiglia declava. This was a real surprise to us. We found it near Yellowstone. But again, another one of these eastern kind of things. This Pepipema. This is going to be included in the new fascicles. So the Lepidoptera Society and the Wedge Entomological Foundation make these fascicles that cover different groups of moths, and they're real technical and research oriented. But Eric, the guy who's doing it, is going to include this record in the new fascicle, which is pretty cool, because it was just kind of such a shock to him that it's occurring here in Montana. And then we've also got these like weird, right? Like you see this map, you're like, certainly it's got to occur somewhere here, right? But in our part of the neighborhood is more of a northwest thing. We found this on MPG Ranch. It's kind of a riparian species that eats deciduous shrubs and trees. Another one of these lithophony that, again, is kind of like this Northwest or Pacific Northwest thing, right? And we found it, these are juniper feeders, which is interesting. And so we found it over in the Pryor Mountains. And interestingly enough, booger eye is named after Jim Truebridge's cat. Apparently it was named Booger, I guess. So, interesting little tidbit there. And then this Eupsilia fringata, again, another one of these California, Northwest species that we're starting to see. And the common theme of all these moths that I'm showing you is they were really partly known in Montana, but their moths have a really distinct composition. So you have spring flying moths, summer moths, fall moths. These are really early flyers. And so historically, not a lot of people were out there in March and April, right, looking for moths. And so we find this in April. In fact, I start mothing in about a week. I go down in March. Another one of these lithophonies. So you can see this is sort of a northern tier species, right? And we've been turning it up in Montana. Another one of these kind of eastern but sort of northern tier species. This is another one that's going to be in that fascicle. This is the typical colored morph. That's the most extreme one, and it's the farthest west that the authors of the fascicle have seen, and we see them on the regular on MPG Ranch. And so that morph is going to be in the new fascicle. And then again, some of these northern tier species kind of, we got this weird pocket here. But the point of showing you these slides is what we're really learning is, one, we really don't know much about moth distribution. Two, Montana is really uniquely situated in the sense that we're getting these Southwest, Great Basin, California influences. We're getting the Pacific Northwest. We're getting the Midwest and East, and also the Northern Tier. And I imagine that's probably true of these front range kind of states, like even Wyoming and Colorado. But we're really starting to see, we're drawing from all these 4 sides. And it's such a big state with a variety of habitats. it's pretty exciting. And so now let's move on to the micro moths. It's an artificial grouping. It's usually been based on size, so these are really small moths, the macro moths are big. But you can, some people say it's phylogenetic, so you can think of micro moths as more primitive, macro moths as kind of less primitive. In fact, the largest macro moth in the world is the wood moth out of Australia. It's a cossid, pretty primitive moth. It can weigh up to 30 grams and be about that big. It's huge. So it's sort of this artificial grouping, but I really, really enjoy these micro moths. As you can see here, I think they're some of the most beautiful moths out there. They just come in all these spectacular patterns. We've sent thousands and thousands. The tricky part about these guys is a lot of the taxonomists that work on these things, they won't give you a name unless they dissect it and look at the genitalia. They'll be like, oh yeah, I think that, yeah, you're probably close, but they'll never come in because a lot of these things have to be dissected to really tell what the species is. And so if we were to consider the number of micromoths that we actually have identified and added to that other list, we're up to about 2,000 species. And the general rule of thumb is let's say if we have 1,400 macromoths, most entomologists would agree that you're probably going to have at least twice, maybe 3 times the number of micromoths. And so our initial estimate that Montana probably is home to 3,000 or 4,000 moth species, it's pretty realistic, pretty amazing in terms of diversity, especially when you compare it to like butterflies, right? And we'll talk about that a little bit later. And so I really enjoy these guys. I spread these guys. I really enjoy working with them. Here's just a picture of one of these cool micromoth spread. And again, you'll see a common theme here, right? This interjectio Columbia is mainly just considered a California thing. I see it on the regular in June and July on MPG Ranch. This Evergestus consimilis, again, like everybody thought it was just a southwest species. We find it in the Priors. And this was a really rare find. It might only be one of the few known specimens that we found in the Priors. And we have several specimens of this Glaucodontia. And then just a few others, right? So you can see, and micromoths are really poorly known. There's tons of undescribed species. Some of them you can't even really identify. But you know, we're filling in the gaps. Like this one only had 2 dots, right? And now we've got it out in Montana. Again, one of these northern tier species, right, that we've now got over in like Medicine Lake area. And then this was a real humdinger. This is a bad picture, but so this is a really exclusive. Like when we sent this to Brian Soltons, he's a crammer. He was just like, man, this blows my mind. It's like it's primarily known as like this southeast species. And it's got a cousin, a relative down in the tropics, and it feeds on Richardia, which is known as like Mexican clover. It's like kind of a tropical plant. And so we're like, what? So we almost suspected this is like an import of some kind. But we found it on state land just down the Bitterroot, by the Bitterroot River. So we're really kind of like blown away by this one. We don't know what to think of this one. But again, the point is you can see with the macros, you know, we're getting these influences from all over. And we're not doing anything special, we're just out there looking for them. But we're really expanding our knowledge of moths. And so we don't really do this alone. In the last couple of years, we've had some folks who have been interested. Josh has done some great work for us out in the Sydney area. Jesse, last year, out of Dillon. This was a really great record, and we'll talk a lot more about this. These Chesiodioides, they're another one of these species that are only known from like California. This one has ranged up in some records in, okay, I'm gonna go. And so Alyssa, you know, they've just done a great job, and what it does for us is it fills in these gaps and provides records that maybe we don't have in the counties for some of these things. Nathaniel over at the Helena Forest has done some stuff for us. Ron over in Trout Creek turned up some great moths. And then, of course, George, and we'll talk about George. George has now talked me into maybe expanding things into North Idaho because he lives in Sandpoint. Yes?
Do you think that some of that research has an
Yeah, like the surveys, yeah. Yeah, like Chuck would say, this what we're doing now should have been done in the 60s. And there has been some folks that were like Frank Lamont and Ferguson and some of these, taxonomists, they did come to Montana, but they generally would, like in the 60s, but they would stay at like a lodge in the Crazy Mountains, and they would just kind of work in that area. Cook in the 30s was a state entomologist that did some statewide work, but nothing as systematic as like what we've done.
But how about all those other states?
Oh, yeah, well, yeah, we've got a lot of interest in people wanting us to figure out a way how they could maybe implement, our model in other states as well. Because certainly, and like I said with George, we may end up doing that with Idaho. And then, the other natural progression would be down into Wyoming. Because I'm sure we'd see really similar stuff we're seeing in Montana in Wyoming, given kind of our same topography and habitat. And, so, yeah, George has got me in a, he got me. George, I guess we're going into North Idaho this year. And then of course I talked about Marion, and Marion has done just a wonderful job in in the Billings area in south central Montana of engaging folks that help her out. You can see some of our volunteers. I don't know why this is on a timer. All the folks that she's kind of engaged. We work with a lot of agency partners as well, and things. And so we're just having a lot of fun. And to her credit, she's done a great job, and I've slacked a little bit, but I kind of chalk that up to the fact that I'm traveling around the state as much. But I would love to be more engaged in our local area and our community, in getting folks like you and other folks involved in what we're doing. And so excitingly, I'm almost done here. We've turned up some really exciting stuff, and so Montana is now home to 2 published new to science species. And the first is this Protogygia priorensis. And it came from the Pryor Mountains. Marion collected it at Petroglyph Canyon down there, where it's one of the few areas that have these kind of open sands, a really unique habitat. She turned this up in early May, and we were just kind of mystified and stumped. We didn't know what it was. So we sent it to Lars Crabo, who, interestingly enough, Lars is a radiologist that is retiring this year, but he has spent his whole life, since the last 25 years, studying moths, and he's described like 70 species or something. And he can dissect them and do all this stuff. So he did the description and stuff, and he was kind enough to let Marion join in on the co-authorship of this, but priorensis means from the priors. And so Montana, that's our, there you go, brand new to science, is our very own moth, means from the priors. Yeah, it's pretty cool. It's really exciting. And then, a year after she discovered that, I was in the Pryor Mountains again in April. Like this was like the middle of April, freezing. Go to my trap in the morning and I pull this moth out. And there was 4 specimens out of 2 different locations, and I'm like, I could get it narrowed down to Chesiodioides, right? But it just didn't add up to like any of the described species. And so we sent it to Cliff Ferris, Cliff is the geometric moth expert in the country. He dissected it, sure enough, brand new species. And so he ended up naming it Chesiodioides montanaensis. So it's from Montana. And we've only got 4 specimens of this. I went back to try to get more, but struck out last year. But pretty cool. So we've got 2 new to science species. Now in 2021, before we even found those, I was near Rhonda, and this one's an enigma. We just have 1 specimen of it, and it's a Glossina species, and it's an enigma because I collected this in like around Labor Day in September, and Glossina are generally like early summer, late spring, early summer flyers, and so it was like really just weird. And so I've been back, I think 5 times to the same site. So I thought, okay, I'll go back in late August one year. Went back, nothing. So then we're like, okay, maybe we're not, at the end of the flight period. So then we're thinking, well, maybe it's double brooded. So I went back another time in like June during the typical flight, nothing. Then back again later in the fall, and still nothing. So it still remains undescribed. Cliff dissected it, it is new, but it's undescribed, and because we only have one specimen, we can't really describe it. But this is, if anybody was curious, this is a male genitalia, by the way. So they take the abdomen. That's how they kind of determine species. And then we've also got these two eupathecia, which again are from the priors, that we think are new. They're currently with some experts. Again, we only have, and then the micromoths is a whole other thing. We've probably got lots of undescribed species there. So anyway, really kind of cool. Not only are we vastly expanding the knowledge of the distribution and diversity in Montana, but we're also contributing new discoveries to science, which is kind of cool. And so just to wrap up, like, why do we care? Why should anybody care about we're going around and figuring out which moths fly where and this and such and such? And the first thing is, there's three big things, is that moths are inextricably linked to plants throughout all their life cycles. So they lay eggs on plants, the caterpillars eat plants. In fact, they probably spend more of their life as caterpillars eating plants than they do in any other phase. And so, some can be pests, right? So some can have economic impacts on things. But by and large, the vast majority of them are out there munching on our native plants, right? So they're kind of an indicator of, like, healthy plant communities. If you got a lot of diverse plants, you got a lot of caterpillars out there munching away on. And the second big thing is that the adults are pollinators, right? And we kind of knew this, but it wasn't until the last 10 years that this, and I'll talk about it in a minute, this meta-barcoding thing has really allowed us to kind of unlock the extent that they are. And then this is just a few examples of studies that have come out in recent years, and I just kind of was like, we're seeing now that they're probably more important in pollinating food production. Systematically overlooked. For 150 years in Switzerland, they were just like, no, red clover is pollinated by bumblebees. Well, they did a study and found like up to 30 to 40% of the plant interactions coming from moths and potential pollination, excluding the significant component. And so what all this means is that we're missing half the story, right? Certainly there's no doubt bees are absolutely crucial, but moths more and more are becoming known as playing a vital role in these ecosystems and these pollination networks. And I'll show you a little data. So this metabarcoding, right? We can take pollen. So this is all pollen on the proboscis, and this is pollen on the moth. And so we can sample this, send it to the lab. They can metabarcode it, and we can get pretty close to like genus, sometimes species, of what plants that pollen is. And so we did a little study on MPG and some of these really impacted areas where they're trying to restore. And I collected, you can see like 300 moths and then about 225 non-moths. So we wanted to compare the nocturnal and diurnal things. We're currently working on it, and I'll just show you a few slides. You can see, and the point here is like we found almost twice as many plant families on the moths as we did the non-moths. And then in terms of genre, it was kind of the same thing, 97 on the moths, and then 57 on the plants. And this is I love this figure. The guy that made this was like, well, there's nothing here. There's no pattern here. There's nothing here. And I'm like, no, this is beautiful, because what it shows is these are all moths and bees, and what it shows is this complementarity, right? So the moths and the bees, they're visiting a lot of the same plants, but also the moths are pollinating things that the bees aren't, and things like that. And so what it really shows to me is like how important moths are to the resiliency of pollination networks and providing that resilience to them. And we did a little work on this rare plant in the Pryor Mountains, and pollinators were unknown. We turned up some evidence that moths might be interacting with them. And then the third thing is they're important food resources. Thanks, Christy. Nice picture. Most bird families rely on caterpillars during the breeding season. Grizzly bears, even some of you might know about the army cutworms that go up into the talus slopes in the summer, and the grizzly bears eat them. And then of course bats, right? And so basically what's happening is the moths are taking all this energy from the plants. They're pollinating plants too, but they're transferring that energy all the way up into these higher trophic levels, even into grizzly bears, right? And so that's why I argue that they're really ecologically important. And so you add that all up, you take these three things plus the fact that there's staggering diversity, right? They outnumber butterflies 9-1. And in bees, they outnumber bees quite a bit too. They're pretty well studied, right? We can look at most moths and, you know, figure out what it is. And then they're relatively easily sampled. Makes them superb ecological indicators, right? So if you're a natural resource manager, and I'm happy to see the state wildlife action plan now is starting to consider insects and things in their plans. And so if you have a really diverse, healthy ecosystem, you probably have a really diverse composition of moths and vice versa, right? So they can be these barometers of environmental health. And then really quickly, I'll wrap up the future. So, you know, we're going to go back, sample all the counties. This is really important. We're currently now in the process of engaging with the Heritage Program. We've shared like 25,000 records with them. They've appended it all into their system. My colleague is now working on about 300 moth species that she's doing, like the kind of the species pages and the habitat suitability models and things, to provide that information that managers or the general public, whatever, can then download and look at reports and things. And so we're trying to bring that data home, if you will, and make it accessible. And then we want to increase citizen science engagement. You know, we'd love to get, if anybody wants a trap this summer, just let me know. I'll get you trained up. And then like I said, we're drinking out of the fire hose. We need to kind of catch up, and we want to do some publications, and eventually, you know, we're going to produce a big checklist. And then the biggie here, and I'm working with, we want to try to like do more pollination data, because we want to try to really change that narrative. That we need to include moths, right? We're missing half the story, so we really want to get more scientific evidence to that. And so anyway, with that, here's some acknowledgments. And how late am I? Okay, good. And then if anyone wants to contact me and they're interested or whatever, just email me. I'd love to take you moth in or get you a moth trap or anything like that. But thank you so much. And I'll take it. Thank you. You have a question?
Are you doing questions?
Absolutely.
So,
I think so, right?
I can make it quick. Sorry. We moved up into Stevensville on some old, like, grazing cow property.
Yeah.
And we're trying to encourage, like, the local toad, western toad population. So we built a few boxes with the black lights around our property
Yeah.
for them to have a place to come eat.
Oh.
But in the morning, we have a lot of moths that are hanging out there, very unique moths. Are we doing more harm than good for the moths? Kind of.
I mean,
We love toads, but we also love moths.
I mean, I would probably say no. I would probably say no. I mean, the fact that you're trying to, like, you know, help western toads, which, you know, is a cool thing.
We are planting a lot of, like, pollinator plants and stuff, but how do you know what, like, the good part and bad part is as far as native species go?
Yeah, I mean, I guess that's a personal
Okay.
ethics kind of line there. I mean, to me, it's, you know, maybe there's things you can do, maybe turn the black light off every once in a while, you know, or things like that. But, you know, it's kind of been known, like, you know, Oppler and Powell, they often said that it only takes 2 eggs per female to survive, to then go reproduce, to maintain a stable population of moths. And so if you use that logic and extrapolate, like, there must be, like, a lot of moths out there. You know what I mean? Like, a lot. I mean, the repeated sampling in one place, but how many moths can a frog actually, or a toad actually eat? Right? I mean, it's not like they're going to be, like, eating, like, 10,000 moths in a night or something. You know what I mean? And so the impact that you're having on the moth populations is probably pretty small. And the fact that you're trying to help western toads, to me, is probably outweighs it a little bit.
So balance.
Yeah.
Turn them off every other night.
Yeah, there you go. Yep, that's what I said. Yes?
During your gathering, have you had any consideration or suspicion that there are invasive species?
So have we documented, yes. Yes, we have. We've turned up, there's one called a Hecatera disodea. It was introduced, it's kind of from like, sort of Iran and that area, and it feeds on like wild celery and stuff. And it got accidentally introduced in like the Salt Lake area, I think in the 80s, and then it started turning up in the Pacific Northwest, and we're now starting to see them here. I don't know how much of a true impact we have. One of the things we have been interested in is there's another one that these biocontrols that we're starting to see. So there's another one called Tida luctuoso, which was introduced as a biocontrol for field bindweed, and we're now starting to see it in western Montana a little bit. And so yeah, there's a little bit of that going on for sure. I'm not sure what the impact is. You know, I mean, some people would argue that like army cutworms are kind of, I mean, they're native, but they might be an invasive species because they can cause real serious damage. And I know a lot of farmers, spray for them and things. But yes, we're starting to see that a little bit, and we'll probably see that more and more. Interestingly enough, the spurge hawk moth, which eats leafy spurge, the roots, it was introduced in Sydney in like the 70s. But like apparently they introduced these biocontrols and nobody ever tracks them. because there was like one dot in Montana before we started this project, and now we've got them all across the state, and so we've been able to see some of that too, of like, where are these biocontrols, how have they expanded? So yes.
Do they interbreed and then therefore every day new species are being created?
There is some species that have been known to hybridize, but a lot of it is just being undiscovered, right? There's a lot of collectors that they estimate that there's about 165,000 described butterflies and moths in the world, and some of these taxonomic experts believe that could be more like 500,000, just because there's so many collectors. Like Chuck got like 300 drawers from a guy that used to live in Laramie, and he was a nuclear engineer or something, and he just loved mothing and spreading, but he never identified anything. And so I'm sure his drawers have got undescribed species that people just haven't seen yet, right? That are just kind of unknown. But some moths, there has been some hybridization, but by and large, they just haven't been discovered yet. Let's go. I think you had your hand up first. Sorry.
What's the proportion of the diurnal and nocturnal? So I know a few that you mentioned. I think it sounded like we're diurnal for the rabbit brush and stuff. So
Yeah. So I'd have to guess, but it's probably like 95 to 5. There's probably like 3 dozen diurnal moths that possibly occur here versus the 1,300 or 1,400. But most moths are diurnal. Or I mean nocturnal. Yeah, sorry.
So then along the lines of hybridization, isn't that why you study the genitalia? Because they're not necessarily interchangeable between species?
Exactly.
So
Right.
then that's how you know that the thing that has a little bit of a curve and other things a little bit flat isn't like some attached here looks unattached here. It's still a completely different species. It's not just a variation on the same one.
Right. Exactly. And that's where the genitalia, yeah, exactly. Yes.
I'm curious if there's been a lot of talk of insect decline across the board. Are there species that there are records of 10, 20, 30 years ago that you're not finding in places?
Well, that's a very interesting question, and we have not got to that yet. So as part of that, developing a checklist and things, so actually in 2 weeks, we're going down to Bozeman, to the Montana Entomological Collection, and we're going to look at what they have there and transcribe their records, so that eventually we will be able to have a data set that we can then look at, right? Because a lot of that collection came from Cook in the 30s, and there's some from Franklin in the 60s and stuff, and so the material that they have there, we should be able to then look at some of these sites and see. We haven't really noticed anything like off the top of our head yet, but Cornell also has all of Frank Lamont's material from the study he did in the crazies in the 60s, and we've begun transcribing that. So eventually, yes, we would like to do that, and then hopefully our material in another 30 years, whatever, will be able to be used even more to try to tell that. But it's a long process going through some of these drawers. It's like, oh my God. Yeah. Yes, way in the back. I'll get.
This might be a silly question, but I'm just wondering, like, at what point, like, can you say, okay, we've discovered, like, every species of moth in Montana? Like, is that a realistic, you know, or, like, can you get to that point?
That's a great question, and I'm not sure if there's a good answer for that. I often just say that when we, I talked about the different compositions and there's spring flyers and summer flyers and stuff. So to me, and a lot of these things are kind of habitat, like, it depends on where their food plant is and stuff, or whatever. So I often answer that by saying when we have sampled every habitat in every season, I think that's a point that we could be like, okay, we feel pretty good about this. Like maybe, it's like that sampling curve, right? Like it's going to plateau and maybe we'll find one or two more, but I think it's going to really take that, honestly, is sampling each county in May, June, July, August, and these different habitats. So it's going to take a while, which is good because I, plan on doing this till I can't, so. Yes. Oh, sorry, I missed you. I'll get to you in a second.
Oh, am I next? Okay. Is anybody collecting caterpillars for these units? Which I know that would be a different preservation method, probably alcohol.
Yeah, probably alcohol. I'm, Dave Wagner. He's out of the University of Connecticut, and he's a really kind of a titan in the LEP world. He does a ton of work with caterpillars. And in fact, he's been doing some work in Idaho, I hear, with the BLM and stuff. But no, that's a whole other ball of wax, right? Because a lot of these moths, like Marion's going through, like with the Heritage Program, and part of her duty is to look for host plant associations. And so now she's in this genus called Uzoa, which is one of the largest moth genuses we have. We think that they're climbing cutworms that feed on grasses and this and that, but most of their caterpillars are unknown. We don't know what they feed on. So that would be something that would be badly needed.
I'm thinking about barcoding of,
Oh yeah, barcoding is always a good one too, yeah. You can do that, yeah. Don't tell them we're going to start a Montana caterpillar project. Yes, sorry, I got you.
Yeah, what would you say is particular about the prior mountains, all these?
All the unique, I think, part of it is that it's our only desert habitat, so it's got a really unique, and there's a ton of endemic plants there and things. And then it kind of sits at this nexus of 3 different ecoregions, but I think, and this is what they theorized in the paper, and we sort of support it, is that it was unglaciated. Right, so it may be one of those areas, right, where some of these relic populations could still exist. It's just a really, really unique area in terms of habitat. But then I think that the fact that it's been unglaciated, I think, is really, it just hasn't been touched by these environmental and geophysical processes over thousands of years that maybe have allowed these populations to persist. Yeah. Anybody else? Thank you so much.
All right, thanks everyone so much for coming. I think it just goes to show that a lot of people just don't look at certain things, and you can discover so much by just being the one that takes the time and has the focus to look. So with that, thanks everyone, and we'll hopefully see you at the next one.